Hi folks, welcome back. Today in this video I want to move on to discussing the next process of molecular biology, that being replication. And let me start by reminding you of what that is here in this slide. This is a slide that I've used before, and this is the complete version of the dogma of molecular biology, the one the evolutionists don't want you to see. And as a reminder, here's the version they teach everyone in just about every science class. The incomplete one, and as I've said, this is intentionally and strategically left incomplete, because once you learn the complete version, you realize that the dogma of molecular biology is impossible to explain scientifically. So now let's get back to the complete version. And as I've already explained numerous times before, this one is impossible to explain scientifically. So I won't go over that again. But what we see here is that the process of transcription creates RNA from DNA, which we already discussed in a previous podcast. And the process of translation creates proteins from RNA. And we've already discussed that one too. And the process of replication creates more DNA from existing DNA. And this process there, the replication, is what I want to discuss today. Replication is the biological process by which the DNA molecule is copied. It's the basis of, and it's a requirement for, the growth of eukaryotic organisms through cell division, which is called mitosis, and the reproduction of eukaryotic organisms, called meiosis, and the reproduction and or growth of prokaryotic organisms, like bacteria. Essentially, life can't grow or be passed on without DNA replication. And what I'll describe in this video is DNA replication from the perspective of eukaryotes, particularly us humans. But know that a similar process exists in the prokaryotes, like bacteria. DNA replication is part of the cell cycle. The cell cycle includes all the phases that a cell goes through prior to and in order for it to divide into two daughter cells. For an organism to grow and reproduce, you must have cell division. That's how all the genetic information is passed on to every other cell in an organism, as well as the cells in the offspring of an organism. All the same genetic information is required for all the new cells of an organism, as well as all the cells of the offspring of an organism, because DNA is what controls and orchestrates everything about life. Without passing along DNA, as is, life runs into a dead end. Therefore, all the phases of the cell cycle and everything that happens during all those phases are essential to pass on life. And in this slide here, I'm showing you an illustration of the phases of the cell cycle. The cell cycle is broken down into phases called G0, G1, S, G2, M, and C. Different events and cell functions occur during each of these cell phases, from essentially a resting phase in G0, where a cell is considered to just be performing its normal day-to-day operations, to M and C, where a cell physically divides into two identical daughter cells. M stands for mitosis, and we're talking about right there, and C stands for cytokinesis, which is that guy right there. Basically, mitosis is the process of cell division. Mitosis, or cell division, is tied into the cell cycle because it's one of the phases of the cell cycle. Mitosis is further broken down into 5 phases called interphase, prophase, metaphase, anaphase, and telophase. Cytokinesis is the final act of mitosis, where the cytoplasm of a cell divides into the two daughter cells. And I'm certain that most, if not all of you, have heard the term mitosis. Anyone who's had a biology course in high school has learned about mitosis. Mitosis creates two new daughter cells from one parent cell, each of which contains the identical genetic information that the parent cell contained. But before the DNA is divided up evenly into the two new daughter cells, it all must first be replicated. And that's what happens during the S phase of the cell cycle. And that's this guy over here, right? And this process is what I want to focus on now. During S phase, the chromosomes and all the DNA they contain are replicated or copied. Now, once they're replicated, each duplicate copy of the chromosome is called a chromatid. Each chromatid remains attached to its sister chromatid, the other copy, via a protein called a kinetochore. Each chromatid has its own kinetochore, which is attached to a region of the DNA called a centromere. The kinetochore binds to the chromatid via a specific DNA sequence of bases within the centromere region of the DNA. This DNA sequence is essential for the kinetochore protein to bind the DNA. So here, if they're showing you this green copy here, is copy 1 of a chromosome, and let's say it's chromosome 1. There's the other copy. So each of those will replicate, and this would be a chromatid, and that would be a chromatid, and they are attached there in the middle by that kinetochore. But now let me get back to discussing DNA replication itself. DNA replication is truly a marvel. Consider that I previously explained how the human genome contains 3.2 billion base pairs of DNA divided up into our 23 pairs of chromosomes, or the 46 altogether, right? That have undergone an extensive amount of coiling and condensing. Every single tightly packed and tightly wound DNA base pair must therefore be unwound, made accessible, replicated, separated, and then put back together again into the two new identical sister chromatids. And not only that, all this happens incredibly quickly. The overall length of time it takes to complete the entire cell cycle varies depending on the cell type. However, on average, DNA replication during the S phase is completed in only a few hours. And then the M stage, or mitosis, is typically completed in only about an hour and a half. And you think you've had a rough day, right? Imagine having to orchestrate and keep track of all this and have it completed in only about half of one workday. Yeah, now that's a busy schedule. To accomplish this amazing feat, DNA replication begins at specific locations called origins. The unwinding of the DNA at the origins creates a structure called a replication fork, where the DNA synthesis occurs. And if you look at this image here, I'm going to give you a rundown of this illustration of everything's going on. But if we're talking about the replication fork, we're talking about that right there, right? Multiple origins and replication forks occur simultaneously during DNA replication. The job of actually copying the DNA belongs to an enzyme called DNA polymerase. And that's them, one there and one there, DNA pol. To create a replication fork and allow DNA polymerase to access the DNA and synthesize the new strands, An enzyme called helicase both unwinds and separates the two strands of DNA ahead of DNA synthesis. And there's my helicase right there, separating the two strands. Helicase does this by breaking the hydrogen bonds between the base pairs. The activity of helicase causes the DNA to become overwound because it introduces what are called positive supercoils. It's kind of like winding a rubber band up too tightly. So to overcome this problem of positive supercoils, enzymes called DNA gyrase and topoisomerase relax the DNA by introducing negative supercoils, like spinning it back in the other direction. And just for the record, topoisomerase has long been my favorite sciencey word. I just love topoisomerase. And by the way, these are those enzymes there that I'm talking about. So it accomplishes this feat by cutting the phosphate backbone of the DNA, or severing the covalent bonds, and allowing the cut strand to slide or spin around the intact strand until the tension created from being wound is resolved. And then it seals the break it introduced in the phosphate backbone by recreating the covalent bond that it severed. Now, once the two strands are separated, single-stranded binding proteins bind the single strands of DNA that have been separated at the replication fork and prevent them from reannealing to one another. So that's what I've got here. Those would be your single-strand binding proteins. So they can't, and they do their job so that the DNA can't close back up again too soon. Now, once the DNA is unwound and the two strands are separated, relaxed, and secured, each strand of the double helix acts as a template for the replication of a new complementary strand, thus creating two double helices from one. This process is therefore considered semi-conservative, since one strand of the new double helix was pre-existing, while the other one was newly synthesized. But DNA synthesis is not only semi-conservative, it's also bidirectional. In the previous videos, when I was talking about DNA structure, I explained how each DNA strand runs in the opposite direction of the other. So this creates a problem for DNA synthesis. The enzyme that synthesizes the new DNA, the DNA polymerase, only works in one direction. It only works 5' to 3'. Therefore, one strand will work just fine for this system, while the other one won't. The other strand is essentially going the wrong way for DNA polymerase. However, all is not lost. DNA synthesis still proceeds in a 5' to 3' direction on each strand because two different mechanisms exist for replicating each new strand. The strand that is replicated continuously, or the one that's in the right direction, is called the leading strand, and that would be this guy I'm showing down here on the bottom. DNA replication occurs uninterrupted on the leading strand in the 5' to 3' direction as it's going towards the replication fork. DNA polymerase moves along the existing strand in a 3' to 5' direction to synthesize the complementary bases on the new strand, creating it in a 5' to 3' direction. It reads the bases on the existing strand, grabs the complementary DNA nucleotide that binds to the existing base, and adds that base to the growing DNA strand. It then moves to the next base and repeats this process over and over and over until the new strand is completed. And I guess you could say a good analogy of this synthesis on this strand is like flowing downstream going with the direction of the current in a river. But now the other strand, this one is called the lagging strand. So that would be this guy I'm showing up here on the top. The lagging strand actually grows in the opposite direction of the overall movement of the replication fork. And continuing with our analogy in a river, these fragments are synthesized by moving upstream, going against the direction of the current in a river. The lagging strand is synthesized discontinuously because it must be synthesized in fragments that elongate away from the replication fork. This happens as a protein called primase, which I'm showing you right there, creates and lays down short DNA primers along the DNA. And I'm giving you one example here of one base. These would typically be 20 to 30 bases or somewhere in that range. 15, maybe 15 to 30, somewhere in that range. Anyway, they put short RNA primers along the DNA. And then the DNA polymerase uses these RNA primers as its required starting material to synthesize a new DNA strand in shorter fragments running away from the replication fork. So as you can see here, this guy's moving this way. These discontinuous fragments are called Okazaki fragments. As replication of an Okazaki fragment completes, DNA polymerase moves ahead, or upstream in our river analogy, to the RNA primer and removes it. So after he did his job here, DNA polymerase would come up here and remove that RNA primer. And then another protein, DNA ligase, then attaches the shorter, separate fragments into one continuous DNA strand by creating the covalent bonds needed between them. So that's what I'm showing you. Ligase here would come in there and connect each of these fragments by creating the covalent bonds between them. In addition to the proteins I just mentioned here, to replicate the DNA polymerase utilizes the assistance of a host of other proteins and RNA molecules. In fact, the complete set of proteins involved in DNA replication is collectively called a replisome. With all these proteins working together, along with several more that I haven't mentioned here today, the DNA replication process moves along the DNA like a zipper. Only this is a technologically advanced zipper. First, it functions as the slider of a zipper, which opens the DNA at the origins as it advances to create the replication forks. This opens up the DNA, or unzips it. Next, it follows along each half of our unzipped zipper and creates a new strand of zipper teeth to pair with the two that already exist. Finally, it follows along each of the two newly created zippers and again functions as the slider to close them back up again. This results in two closed zippers where there was once only one. DNA replication is truly an amazing process. And just like I did with transcription and translation, I just gave you about a semester's worth of material from molecular biology in about 10, 15 minutes or so, whatever it is. And I left a ton of stuff out, from its orchestration to its efficiency, to its problem solving, to its effectiveness, its choreography, its speed, whatever, all the above. DNA replication is truly a marvel. When you fully study the process of DNA replication, it's almost beyond belief that it even is possible in the first place. And what's even more amazing is all the information needed to make it all happen comes from DNA itself. And these molecules that I discussed today are more examples of the 300 or so molecules that I've discussed previously that are needed for the life-essential processes of molecular biology. All these molecules are critically important for these processes, and therefore critically important for life. With any of these molecules missing, critical parts of these processes won't happen. And if any of these steps in the process don't happen, life cannot happen. Therefore, as I've already explained, all of these molecules have to be here at the same time, functioning together, or life cannot exist. They cannot accumulate gradually over time because they're all needed together from day one, which means they simply cannot arise via evolution. And as I've reviewed in previous videos, this again is also irreducible complexity to the utmost. All right, well, I'm going to end this video here, but come on back for the next one where I'll provide you with more evidence from DNA replication that shows it could not have arisen via evolution. And if you like what you hear, give me a thumbs up and subscribe to my channel. Thank you very much for watching, and as always, we'll leave the light on for you.